Hello, good afternoon. Like she said, I'm Brett Jones. Today I will be talking about polymorphisms at the DRD2 gene, how they affect dry matter intake and weight gain of heifers fed tall fescue silage. So just a quick outline of what I'm going to be talking about today. A short introduction about the importance of tall fescue toxicosis. Then I'll explain a little bit about the cattle genotyping I used on my research. I'll then cover my research objectives, some experimental design, then get to my results and conclusions, and then I'll take any questions. So just an introduction. Tall fescue is one of the most important, or most popular, in my opinion most important, popular perennial cool season grasses in the United States. It is grown on approximately 32 million acres nationwide. It has good tolerance to overgrazing, environmental conditions such as drought, heat stress, and cold temperatures, and then also low soil fertility and poor drainage, and then also has some insect tolerance. So tall fescue toxicosis is a result of tall fescue which is infected with endophyte. I have a picture here of microscopic level of the endophyte between the plant cells in the plant. There are three different tall fescue endophyte associations. There is toxic, endophyte-free, and novel endophyte infected. So for the toxic endophyte infected tall fescue, it produces ergodalkaloids, the most common of which is ergovaline. These ergodalkaloids cause many negative effects on livestock, but also help the plant to be very persistent. The next endophyte association is the endophyte-free. Obviously, there's no endophyte, so it does not produce those ergodalkaloids, which then results in poor plant persistence, but no harm to the livestock with this type. And the third is the novel endophyte association. This association produces either none or less ergodalkaloids, but still provides the plant persistence and no harm to the livestock. Tall fescue toxicosis causes many negative effects in livestock. The first two I have listed on here, decreased weight gain and reduced intake, are the two which I will be covering a little later. This is a long list of a whole bunch of negative effects I won't read off, but none of them are good for the animals. It ends up costing U.S. livestock producers over $1 billion annually. Agronomic solutions for tall fescue toxicosis are costly, risky, and as well as time-consuming. So for the cattle genotype that I used on my research, it was the DRD2 gene, which was researched by Campbell et al. In 2014. It was research done by the University of Tennessee and the University of Missouri. This gene is a dopamine receptor D2. At this gene, there's a single nucleotide polymorphism that was discovered where an adenine and guanine substitution exists. This gene is important in the regulation of prolactin secretion. So one of the negative effects that I listed earlier was low serum prolactin. Tall fescue toxicosis and decreased serum prolactin. For my research, I used the homozygous animals. So the tolerant animals are the AA and the susceptible animals are the GG. There are heterozygous animals. I'm not using any of them because in the research conducted, it showed that they had intermediate effects with the, so the weight gain. And I decided just to use the homozygous animals in my research. So here I have a picture of two animals. I don't know how well you can see that, but these are two animals on my study. The animal on the left, that is a tolerant animal. The animal on the right is a susceptible. You'll notice the animal on the right has retained its hair coat from the winter. That's significant because this picture was taken in late July, early August. So obviously this animal has been suffering from increased heat stress due to that retained hair coat. Its body condition was also lower, where the animal on the left, the slicker hair coat, obviously better condition, gaining better. So for my research, the objectives were to determine the difference in intake of genotyped heifers fed tall fescue silage infected with either a novel endophyte or a toxic endophyte. And the second one was to determine the difference in average daily gain of genotype heifers fed either Bar Optima or Kentucky 31 ration. So a little bit of my experimental design. There were 4 treatments in a 2x2 factorial arrangement. There was the tall fescue. I used Bar Optima plus E34, which was my novel endophyte tall fescue, and then Kentucky 31, which was the toxic wild type. For my genotypes, like I said, I used tolerant AA and susceptible GG animals. It was a randomized complete block design with 4 blocks, and I used SAS proc glimics to analyze. The tall fescue was harvested in the spring around the 1st of May. It was round baled and then wrapped in tubes. It was allowed to set for a month, and then we tested it for its nutritional value. Bar Optima plus E34 had ergovaline levels at 50 parts per billion and total ergodalkaloids averaging 287 parts per billion. The Kentucky 31 ergovaline levels averaged 405 parts per billion and total ergodalkaloids averaged 2,196 parts per billion. For the rations, I used minimal supplement to make them isonitrogenous and isocaloric. The heifers that were used in the study were raised on 3 different University of Missouri research farms: the Southwest Research Center in Mount Vernon, Missouri, the Beef Research and Teaching Center in Columbia, Missouri, and the Forage Systems Research Center in Linneus, Missouri. The heifers were Black Angus, Red Angus, and a Simmental Angus cross between the ages of 9 and 14 months. These heifers were fed in the Grow Safe feeding system at the Southwest Research Center in Mount Vernon. So the Grow Safe feeding system, for those of you who haven't ever heard of it or been able to see it, I have a picture here. This was taken on my research. So each animal has an EID tag in their ear. When they put their head through the bars that you can see, the bunks shown here have a reader around the edge that reads that individual's ID. The bunk sits on weigh bars, which weigh the feed in the bunk on a continual basis. That weight is then sent wirelessly back to the headquarters, where it is continually collected and able to be viewed and analyzed there. So the layout of the Grow Safe system, there was 8 feeding pens with 2 Grow Safe feeders per pen. We put 6 heifers per pen, giving us a total of 48 heifers. The intake was continuously collected for 70 days, and the heifers were weighed every 35 days. And there's kind of the diagram of the layout. There was 2 barns with 4 pens in each barn. So for some of my results, this is preliminary data just from 2013. This is the animal intake. So what you'll notice here is the Bar Optima animals were not significantly different in their intake. It's what we expected. What was the significant thing here was the Kentucky 31. Tolerant animals consuming Kentucky 31 were not significantly different than those animals consuming Bar Optima, with the susceptible animals consuming Bar Optima or Kentucky 31 being significantly less. For the average daily gain, you'll notice the Bar Optima, again, no significant difference between those Bar Optima tolerant or susceptible. There was, though, no difference in the Kentucky 31, but tolerant animals on Kentucky 31 were not significantly different from those animals consuming bar optima. So in conclusion, GG heifers, the susceptible heifers, had significantly lower intakes when consuming the toxic endophyte infected Kentucky 31 ration. The average daily gains of AA heifers fed Kentucky 31 were not significantly different from heifers fed the novel endophyte infected bar optima ration. And finally, the preliminary data indicates that the DRD2 gene may provide a way to mitigate tall fescue toxicosis if non-toxic rations are not feasible. With that, I will be glad to take any questions. Yes?
I was wondering why you saw 287 parts per billion of ergovaline in your novel endophyte tall fescue.
So it was 287 parts per billion of total ergot alkaloids. It was only the 50, I think, of the ergovaline. Bar optima actually does, it has some of the ergovaline ergot alkaloids in it. Not as much. There is also the possibility that the stand that we harvested that from had Kentucky 31 or a toxic. What I guess I should say is it may not have been a pure stand. But that's why we tested for the ergot alkaloids and ergovaline, was to make sure that it wasn't at a level that was going to be harmful to the animals.
What's your threshold? Did you have a threshold level for total ergot alkaloids?
Not for total ergot alkaloids. I was more ergovaline was more of what I was looking at. But for ergovaline, I think the number is around 200 parts per billion of where you usually start seeing some kind of an effect on the animals.
Did y'all take any of the other measurements to look at fescue toxicosis, body temperature, serum prolactin, any other type of measure?
Yes, we did. Both of those actually. The temperature, I didn't include that. The serum prolactin we haven't analyzed yet. I was just going to analyze the serum prolactin in one big batch at the end of my research. So I don't have that to present, but I do have those numbers, yes.
We have time for one more question.
What do they think the heritability will be? I'm not too familiar with the singleness, the kind of polymorphism. But is it going to be a simply inherited trait?
So it is. It would be just, if an AA is bred to a GG, you're going to get heterozygous offspring. What they did see in the research with the University of Tennessee and University of Missouri was that, there's some natural selection, animals that are, you have the likelihood of having fewer susceptible animals in your herd just due to loss of calves, or just the result of tall fescue toxicosis culling them from your herds.
But the industry believes that tolerance or adaptability occurs from animals that live in an environment and stay in that environment and they reproduce, and over time whatever works in that environment is what you have. But your work kind of says maybe that we're not getting what we think we're getting in tolerance to fescue and its effects on the animal.
I think you do see sometimes where the animals, if that's all they're exposed to, they might become, a little. But at the same time, you wouldn't ever really maybe even know the difference, So it's.